Hello, welcome. This is Dr. James again, Danoff-Burg from the Living Desert Zoo and Gardens, and we are here to talk about session 8, which is all about the ways that we would go about summarizing and representing and statistically analyzing our quantitative data, which is for many of us going to be the majority of the data that we collect, of course. A topic, like today for this session is that would adequately do justice to all of the topics of this would be one that would last an entire year, 2 years. So the material that we'll be able to cover today in our rather limited time together is necessarily going to be mostly referencing you to other places that you can find out more information to be able to make those difficult decisions that you need to make in order to analyze your data with statistics. So last time we talked quite a bit about doing graphing and descriptive statistics, calculating averages and means and maximums and sums and all sorts of things. And we'll go through some of the nuts and bolts of how to do that here today. But the statistical programs that one needs in order to analyze this are a whole group of topics that we're not going to be able to get into too much more than just my quick introduction to what we're talking about. During our time together today, I'm going to be switching back and forth between our PowerPoint and an Excel file and a whole series of websites, and also a Adobe PDF viewer to show you a few other documents that we'll have available to you in the class site. So all of these are a series of resources that are designed to provide you support to be able to make it possible for you to do what you need to do. Coming out of today, you will not have the profound understanding of statistics because that's impossible, but you will have an understanding about what are the ways in which one goes about solving those statistics questions and how to resolve the problems that we're facing. Sound fair? So very often teaching is about expectations management, and this is that process. So we're not going to teach you about all of statistics today. We will dip our toe into the pool of statistics to the degree that's appropriate for a course like this. Statistics is one of my favorite things to do and as a scientist, and I hope that it is just as interesting and becomes a passion for you as much as it is for me. So you may already be familiar with doing statistics. Hang out. We're going to be going through a bunch of things that maybe will provide you some new information. I want to share with you a whole series of approaches as well as a statistical analytical software that you can use to analyze your data that is entirely free. And not only is it free, but it has a point and click or a graphical user interface or GUI interface, if you're familiar with those terms. So there are solutions, and I wanted to provide you some of those solutions today. So without further ado, let's go to the PowerPoint. I'll share that here and make it full screen. So today is session 8, Quantitative Statistical Analyses. And what this means is that our course is not quite done, but we are, after today, 8 of 11 sessions in. Our next 2 sessions are all about qualitative data research, our open-ended questions, our open-ended items. And Alex Ocañas is going to be revisiting us again after a long series of just you and me for these next 2 sessions. And so she'll be leading these discussions for sessions 9 and 10 on qualitative research design and qualitative data analysis. We introduced some of these terms already, but these are going to be a greater and more useful in-depth introduction to these topics. And then in 3 weeks, we will wrap everything up and come up with implementation plans for implementing our social science research. All right, so today, we're going to touch back again and go into a little bit more depth with how to visually summarize our descriptive statistics And I'm going to show you on an Excel file some easy, quick, and dirty ways to go about creating the data that one needs in order to create those graphs, those bar charts and line intercept graphs and pie charts and whatnot that we talked briefly about at the end of session 7. Then we're going to talk a little bit about why statistics is so important and what it means and how we interpret it. Then we'll get into a whole series of discussions about how to choose between different statistical analyses, whether a chi-square, an ANOVA, a t-test, a multivariate regression, a Kruskal-Wallis, a Mann-Whitney, a freak. There's so many statistical tests, it'll make your head spin unless you are a statistician. So I'm going to walk you through a series of tools to make it possible for you to choose between those statistical approaches, which program and which statistical analytical method is going to be most appropriate for what you have and for what data you have to answer what questions you're trying to answer. All right. I want to introduce you to the program R, and I want to share with you a document that I wrote 4 years ago about how to download it and a companion program called R Commander, and then how to install it, how to use it, how to do analyses with it. It's a very nice intro to R, an intro to doing statistical analyses for quantitative data with R, a free program that is a great tool to use. So I encourage you to actually follow up with that and use that PDF to make this happen. We'll talk very briefly about inputting data into R, and then I'll walk you through some statistical analyses using the R program. All right, so that's the agenda for our time together today. I want to refresh 2 main terms that we talked about, or sets of ideas that we talked about last time. When we were talking about interviewing people and doing it in a way that is defensible and well-designed and gives us good data, we talked about anonymity versus confidentiality. And I want to underscore these 2 again very briefly here. Anonymity is when we do not collect the information of the people themselves at all. There's no information that we collect about a person that would make it possible for even us, who are the people speaking to them, to be able to say that person said this thing in the future. So anonymity is sort of the gold standard. It's really a useful thing to promise to our participants. People will feel much more comfortable in being more open and honest and direct with you if you can provide them anonymity. In contrast, confidentiality is when we know who the person is, we know their face, but we obscure their face a bit by not putting their name or their individually identifying information with their responses. So we take that individually identifying information and we put it in one data set, and we put a number, a code number to it. That is the same code number that we put in a separate file that's kept on a separate computer, both of them password protected, that has that code number as well, so that if you have the identifying information file, you can use that code number to link person number 156 to 156 so that you know that it's this person. And the benefit of that, of course, is that in the future, if you're trying to do a pre and post test where you're talking to this same woman that's on the screen, before you do your intervention on using very sustainable cooking stoves, and you want to be able to go back to that same person and ask her that same information after she has been using those cooking stoves for a while, then you would figure out, okay, respondent number 156 lives here, and this is her name, and this is her age. And with that information, you can go back to where she lives, and you can ask that woman again the same questions afterwards as before. That's the value of confidentiality. So you're protecting them, and you're protecting the respondents by not having their individually identifying information in the same file, and almost always never on the same computer. All right, so anonymity versus confidentiality. Okay, last time we talked about a code book and how valuable code books are, and that we need to create code books for our quantitative data. We also create code books for our qualitative data Alex will talk about codes and themes and expand on that introduction that we did last time. And she'll talk about that in these next two sessions, sessions 9 and 10. But keep in mind that the purpose of a code book is to take those textual answers, I am a man, I am a woman, and put them into numbers for quantitative data. The value of that, of course, is that we can produce spreadsheets that look like this. And these are the spreadsheets, and I have a sample one that we'll use today that you can put into your statistical analytical packages, your R, as we're going to use today, and then be able to determine whether group 1 is different from group 2 significantly based on question number 13, for example, as we have on the screen here. All right, so code books are really important, but being able to code our data, our quantitative data, into numbers, into quantities, is the importance of the code book data. It provides us a way to be able to take words or concepts and put them into numbers. And then we can analyze them in statistical packages like R, or SPSS, or SAS, or Minitab, or what have you. There's lots of different computer programs out in the world for doing statistical analyses. R is a free one, it's open source, and so that's one that I'm going to walk us through today. All right, so next up. Last time we talked about the difference between descriptive statistics and inferential statistics. As a reminder, because we're going into both of these quite a bit more today. Descriptive statistics describes the data. So it talks about the averages, or means is another way to talk about what we really are, because there are many kinds of averages, and what most people talk about when they say, well, I'm going to average my data, is really a mean value, because there are other types of averages. But we're counting frequencies, we're totaling up the total number of cattle in one area, for example, so we're summing it. And then we're taking that information to make graphical representations. Sometimes it's like this pie chart that's on the screen. Sometimes it could just be a data table. But however it is, we're summarizing that data and describing it. And that is descriptive statistics, which is very helpful. That does not tell you whether something is significantly different from another thing, whether group A is different from group B in the number of cattle that it has taken by predators, for example. The value of that, or that what you need in order to do that, is inferential statistics, because inferential statistics infer differences between our data, often between group 1 and group 2, right? So examples of inferential statistics, some of which we've already mentioned today, many of which you already know about, of course, are chi-squared tests, ANOVA, as you may have heard. And ANOVA is itself an acronym. It stands for an analysis of variance. Analysis of variance. A simplified ANOVA is called a t-test if you just have two groups that you're comparing. Regression is when you have continuous data, say height or weight, something like that, and you're trying to graph one against the other. And we're going to go into these inferential statistics quite a bit more today. We'll touch briefly here on the descriptive statistics, and I'll walk us through some Excel file, or an Excel file, with some Excel actions that we can do. All right, so there are lots of ways that we can summarize our descriptive statistics. So we're going back to descriptive statistics here. And this is a slide we had last time. We can show it as stacked horizontal bars, as in the image on the screen. We can put it in pie charts. We can put it in line graphs if there's a year after year summary, as in the graph on the screen that talks about population sizes of bears, dolphins, and whales in that area. Similarly, it can be vertical bars, as in the results saying that Grizzly Ridge inspired me to spend more time outdoors. Number of people that agreed to each of those. Here's a question for you, a refresher. What kind of a question or an item is this that we're summarizing? Is it a quantitative or is it a qualitative? First off, quantitative, right? And so within quantitative data, is it a categorical data? No. Is it a ratings data where we're rating things from 1 to 5, most favorite to least favorite? No. Is it a ranking? No. What's the term? It's a Likert scale, because we're going with strongly disagree to strongly agree, right? So on this one, people generally agreed with the statement that Grizzly Ridge inspired me to spend more time outdoors. And you see n equals 93. N equals 93, what does that mean? If you said that it's the number of participants in the survey, you're spot on, right? So that's our sample size, is 93. Okay, so these are some ways to show data. Now, let's calculate some of the things that that data summarized and just showed, right? So we'll talk about averages, our mean, and as I had said earlier, there are multiple kinds of averages. There are means, there are modes, which is the most common data point. So if we look at height, for example, in the United States, the modal average height for women is usually right around 5 feet 4 inches. I'm sorry, I don't know what that is in centimeters. We're stuck on this English system. That is so therefore the number of people who had the greatest, or the group of people who had the most common height would be the mode. Often, and in very large samples, the mode is pretty much the same as the mean, excuse me. So, very small sample sizes, of course, you could have skewed data. Maybe you work with basketball teams. And so the most common mode of heights of women who are playing basketball on your team are going to be much greater than the modal average of the normal or run-of-the-mill Americans, right? So just by chance. And in small groups, our mode does not necessarily overlap with where our mean is. Ideally, they will, but generally, they are very separate from each other. Okay, so our third kind of average is called a median. And a median is if you take all of the data points, say take all of the heights and we arrange them all up in a row, and we have, let's say 11 people, 11 heights, and we've lined them up from shortest to tallest, and then we look at the centermost person. And the centermost person is going to be the 6th person in from the shortest, the 6th person in from the tallest. Right? And so that person is the median. In very large groups, again, our median lines up with our mode, which lines up perfectly with our mean. But in small groups, because of sampling error, as we talked about earlier, small groups tend to have larger sampling error. And so therefore, our mean may be here, but our median could be much greater or much smaller, depending upon who the people were that you brought into your group for analysis. All right, so that's averages. There's 3 of them. The one that most people use is what we call mean. We're going to go to Excel in a moment, and I'll show you how to calculate each of these. These are the formulae that are on the screen right there on the right, where it says equals average, parentheses, B2 colon B11, close parentheses. That says take the mean, because Excel unfortunately uses average instead of mean, but it says take the mean of the data in cells B2 through B11. So it adds up all of those, divides them by the number of them. That's how you get the mean, or the average, as most of us call it. But we can do the same thing to calculate the mode. Substitute mode instead of average, as you see on the screen here. You can also calculate the median by substituting median for what was mode or average with the same data range. Importantly, when you're typing in these formulae, if you like to type in formulae, you put the equals sign in Excel, and then that tells Excel that it is needs to calculate something. And this is the formulae for how to calculate each of these means, each of these averages, excuse me. Same thing for sum, for maximum value, for minimum value, for the standard deviation, which is a way to summarize the amount of noise that you have in your data, right? Which is the difference of each data point from the mean value. That's how you calculate standard deviation generally. So these are all useful things. So now let's go over to our Excel file. So I'm going to stop this share and I'm going to share my Excel file that I have open here, which is a, there it is. I couldn't find it right away. Which is some data that we collected recently with intervening with restaurants in two main cities, Yucca Valley and 29 Palms, which is what YV and TNP are in the first column. That's the cities. And in some of them, in the experimental groups, as you can see on the bottom of the screen here in column B, all of these, we went in and we talked to the staff. We talked to the managers and we asked them to close the top of their garbage cans. Right? Why, you may ask, are people who are doing conservation wanting to talk to restaurants and asking them to close their garbage can lids? Well, we have a program here called the Time to Talk Trash campaign. And what we're trying to do is to get everyone to cover their trash, which is what the link is on the bottom. If you want to go to this website, coveryourtrash.org, you can see the program that we do. And we work across the deserts in the southwestern United States because ravens, which are a predatory corvid, if you know what a corvid is, like jays, they're very smart birds, but their populations are 17 times in some parts of the desert that we live in, greater than what they were only 30 years ago. And that is largely because of all the extra trash that is here in the deserts in which we live, and also extra water that we're providing them, and shelter and nesting spaces. So we want to try to reduce the amount of the greatest, what we call a subsidy, the trash that's being provided to the ravens by covering your trash. That's why. And so we work with those restaurants to try to get them to cover their trash. And we had a bunch of different reasons that we told them why it's beneficial to them to cover their trash. Health codes, health code violations if they leave it open, perceptions by the public, reduced litter flying around because things blow out of the cans. And these are very large cans. We call them dumpsters here in the United States. So we want these restaurants to cover their trash, cover their dumpsters. And so in the experimental group, those on the screen on the bottom right here, or bottom left, excuse me, are the experimental group. These are the ones that we went in and spoke to. The top 30 rows are the control group. And in those two towns, we did not talk to them. We did look at how frequently they closed their dumpsters before we did our interventions with the other restaurants, and then how often they closed their dumpster lids, their garbage, trash can lids at the end, the rubbish bins, and we looked at the difference. So we, in both the control and the experimental plots, in the control and experimental groups, we went in and did a before test. We went back 5 times and we recorded the number of times that the lid was closed. As you can see on the screen here, the first restaurant had its bin closed 3 of the 5 times. The next one had it closed every time. Some restaurants never had them closed. They never closed their lids of their dumpsters or of their bins. And so we did a pre-test and we did a post-test where we went back 10 times in total, 5 before, 5 after, and measured how many times they closed it. So that's the setup for this experiment. And overall, if you look at this, here you can see in the cells that I've typed in the average values, which is the mean. It calculates the mean, not the mode or the median, for the pre-test, for the post-test, and then the difference between the pre and the post, which was the post-test subtracted from it the pre-test. So if there was a positive number, it means that more frequently in the post-test, they closed their lids, possibly because of our intervention of going in and speaking to them. Okay, so if this is a positive number, that means that there are more dumpster lids closed after our interventions than before. If it's a negative number, it goes the opposite. If there's no difference, then there's no difference. And we support the null hypothesis, as we're going to talk about. So as you can see, these are averages in the cells. These are the formulae. Average pretest was 2.69 times the lids were closed when we came by. And in the post-test, on average, 3.32 times the lids were closed. There was a positive difference, meaning that there was an increase in the time between our pre and post. And then we go down to and I'll come back to the maximum and minimum in a second. The pretest, and just looking at the controls, there was almost no difference between these two. An average, a mean of 0.38 times greater post-test to pretest. And if you look at just the experimental groups, just those that we talked to, the average before was 2.93, the average afterwards was almost 4. So it was almost an increase in average closure of their dumpster lids of 1 out of 5. So a 20% almost increase in improvement overall in just the experimentals, which is pretty amazing. So we actually got some very interesting results from this and have concluded that this intervention really does work. And so we're very proud of it and we're continuing it. But all of that is beside the point. What we're trying to show you here is how to calculate averages, which I've created here. And it's the exact same formula as in the averages. If you keep looking at the top cell here, the maximum and the average have almost the exact same formula, just substituting max for what was average. And this will tell me what was the maximum in the pre-test overall, what was the maximum in the post-test overall, and what was the maximum in the difference. Make sense? So one restaurant, I'll give it away, one restaurant went from never closing it to always closing it afterwards. And that was one of the ones that we had an intervention and spoke to them about. It was, they're sort of our poster children for this program, the ones that we are most grateful for. There was, of course, one restaurant that went the other way, that before they closed it maybe 4 times, and then afterwards they never closed it in the post-test. So that's why that -4 overall is the minimum. Okay, so same thing with the controls, same thing with the experimental. These numbers aren't all that interesting, really. Although it is interesting to see that the difference in the experimental overall is the smallest, and the smallest decrease minimum is in the control group of -4, whereas in the experimental, the smallest reduction from pre to post in terms of the number of times that they've closed their lid is only going down 1. So we did find an interesting difference here, and we were very grateful for that. But what I want to show you as well is that you can calculate all those things, of course. You can also substitute sum in here if you wanted to total the number of times that the lids overall were closed. And I'll type that in. I'll put in average, sum, and then open parentheses. And then I will sum the pretest values, all of them, for both sites in the control and the experimental. And so you can see what I'm doing is I'm just clicking, holding, and dragging down to the bottom. And when I get to the cell that I want, I let go, hit close parentheses, and then return. And you can see that the sum for the pretest, as in over here, copy these and put them here, is that. And so then the sum of the post-test, I'll do the same thing, sum parentheses, go to the topmost data point here, click all the way, click and hold and scroll all the way down to the bottom. All of this is pretty rudimentary Excel, but it's also good to be able to have this in our toolkit. So we can see overall that there was an increase across all sites, all restaurants, control and experimental combined, from 159 times the lids were closed in the 60 restaurants that we went to. And afterwards, after the intervention, it went up to 196. That's a pretty good increase, right? So that's how you can do some as well. Now, let's go back to our data. Hopefully that's a useful introduction. But if you wanted to put in another formula here, let me show you another way to go about doing this so that you don't have to memorize all of those keystrokes if you're not comfortable typing those things in. In Excel, you can see on the top of the screen here, there is that FX here. If you click on that, that shows you all of the most recently used functions. Sum, which we just did, average, which we just did, min, that we just did, max, and then there are a variety of more complicated ones, including mode and median and a bunch of other options. If you click on one of these, say average, and double click on it, then you need only highlight the cells that you want to actually average. So instead of typing it in, what you could do is come over to the post-test or pre-test data, click, hold, drag, go all the way down to the bottom. And this time, maybe we want to know the total overall times that the rubbish bins lids were closed. So we're doing all of column C and all of column D, which is the pre and the post tests. Click it over, I let go, and then I hit return or enter, and I'll close that. I'll move it over to the side. And there is the average overall of the number of times that the dumpster lids were closed. Not some, excuse me. I thought I clicked some, but I hit average. So this is the average overall of how frequently the lids were open, pre, post, across all restaurants, whether control or experimental, whether in one community or in the other community. Okay, so I'm going to close that. And now I'm going to stop the share here and go back to our PowerPoint. Now, that is how you calculate those descriptive statistics. Try this with your own data. Try opening up a data file and try playing around with these average, sums, maximum, minimum, standard deviation. If you're interested in the mode and the median, because those are very useful numbers sometimes for different studies, do so. All right, so if that's something that you would want to be able to do in the future, of course, these notes are on our class site, so you can get them from there as well. So continuing on, that's how you calculate those descriptive statistics. So try that out. And if you want to do it now, you can pause the screen and open up your data set and try typing in these things here. Well, we'll take a drink of some tea while you pause the video and do that. Okay, so also just like to point out, we'll continue on now, but this is from the Desert Tortoise Council. So this is the reason why we are doing the Time to Talk Trash campaign, is to try to benefit desert tortoises. And that's why, because ravens are a major predator of desert tortoises. Desert tortoises are adorable and absolutely worth conserving for so many reasons, not just because they're adorable, but because they are important parts of our ecosystem, and we need to keep those beautiful animals around. I'm looking through my sticker there. That's what they look like. That's the tortoise calling out for help. So anyway, that's how you do descriptive statistics. That's how you calculate these things. Okay, so if we have our charts, which we have on our data sheets that we have in our other things, and our other Excel file that I have open as well, let's talk about creating charts. It's very easy to create charts in Excel. And just so that you know, R, that statistical program that we're going to get to, does not have a very useful, or I'd say robust, charting capabilities. So I create most of my charts in Excel because it is such a robust program. So think about using Excel. It's an easy way, if you have access to it. great. If you don't, you can also use Google Sheets Online. It's comparable in a lot of ways, and that's free. Sometimes if you have a Graph, I think it's called, on the Mac, but there are free graphing programs or spreadsheets around. Most spreadsheet programs can create a pretty decent chart. Excel, for the paid version, has really great charting capabilities. So let's create some. All right, so we'll first create a pie chart. All right, so let's go over to our data sheet again in Excel. I'm going to stop the share here, swap over to our data sheet. So quickly that it makes your head spin. There you go. Took me a minute to find it. So we'll create a simple pie chart. So here I've summarized in these cells down here the frequency, the number of restaurants that we went to in each of the 4 kind of conditions, whether it's in one community or the second community, Yucca Valley or 29 Palms, very desert sounding ecosystems, and then in the experimental and control groups. So we summarized all of these. There are 15 pretty much in every one except for the 29 Palms control group. Because we ran out of restaurants there, so we were short one. But if we want to be able to create a pie chart, it is very easy. As you can see, I've highlighted the data, and then I come up to the charts option on the top of the ribbon in Excel, come over to a pie chart. You can see that on the screen here. Click that, and I'll just do a simple pie chart. I click that, boom, it is up and ready for you right there. So you can see that they're almost the same number in each of the 4 kind of conditions, YV control, YV experimental, TNP control, and experimental. So that's an easy way to do that. If you would like to label the charts, if you want to change whether there's a header, whether each of the text descriptors are inside the cell, you can come up here to the top. You see where it says chart quick layouts, and sometimes it might have exactly what you want. So you can just come in and click the layout that you like, that is best for you, best for your project. Maybe we like that one best, and we want it to be in black and white. Or maybe we want it all to be in different shades of red for some reason, because that's relevant, or blue, or the default colors for here. And then if you want to put in a chart title, you just click into the cell, into the box, double click on the chart, as I've done, and then delete it, and type experimental sites in it, and just click off of it, and then you've got a cell, or you've got a chart. And you can copy this chart and then paste it in a Word file or in a document file and whatever you have, and then you have it there. You can continue to modify the colors, you can modify the layout, you can make the pie chart bigger by just clicking and dragging it. How do I get to this thing? Usually you can, I work on a Mac and a PC, so sometimes things get a little confusing. But if you want to make it bigger, of course, you can also just click the overall thing and drag it down that way. If you want to change where it's located in the cell, you can do that. If you want to break it apart, you can just grab it and pull it apart. There are a lot of different options and things that you can do. Play around with your screen. You can change the colors of each of the areas. So you format the data point and change it from that color blue to, say we want that one in black. I don't know why we do that, because then we've lost the text, but that's what we did. And of course you can hit the Control-Z to reverse that if you're on a PC. So that's how you make a pie chart. Very simple. Now let's go back to our data sheet, or our program, PowerPoint show, And there are lots of ways that you can go about modifying this in different ways. Some of the ways that we talked about, changing the color scheme, changing the layouts, the quick layouts as they had on there. There are lots of other options. If you just get in there and play with it, you'll figure it out. Next thing, with the legend, you can change what the legend says very easy in a lot of ways. And here are some ways to do so. You can also just go into the Excel file and experiment. Usually if you go in and you're actively still working in the Excel file, you can just change what this column with the labels say, and the chart will automatically update, depending upon which version of Excel you have and whether you're working on a PC or a Mac. Sometimes it's very easy. Here are some other suggestions and options for how to go about that process. Experiment, you'll come up with a solution. Okay, so you can also change how the charts are labeled. And this is an important thing that I'd like you to focus on, because ideally when we're putting in our title or our label for the charts, we'll call the title is the top, the label are what each of the parts are. We have a clear description about what this question was, what the item was, what we wanted people to say. Sometimes different people label their charts as there was a significant number of people, or people were mostly strongly agreed, and then summarize that statistic of the summary of the two of 77, adding together the turquoise blue strongly agree and then the purple agree of 37. And you can put that into a title. Most people agreed that they felt a stronger connection to these animals now. And then you'll notice underneath that title that there is a N equals 11. And we mentioned this earlier in that N tells you the number of people who participated in the survey or who answered that item. Sometimes people will skip items, and that's okay. But we have the ability to summarize the number of people who actually answered our questions. Make sense? So this is how we can summarize a lot of data, a lot of descriptive data very quickly using pie charts. And so try this on your own, but make certain that when you come up with your final ones, you always say how many people, how many participants you had in your study. People will appreciate that. It'll help to inform how much strength they feel that they should put on your interpretation and the data that you have right there. All right, so continuing on. Now, let's make a bar chart, and we'll make that using that same data in the other program, in the Excel that I'm using. And we can do this, we can do a lot of things with it. We can swap the bar charts. We can put the percent responses on the x-axis, or on the y-axis, excuse me, and then the rating options on the x-axis, and the opposite of the graph that's on the screen for how much people enjoyed the program. And you'll notice that it has the participant number N equals 101 at the top underneath the title. But there are lots of things that you can do. Get in here, play around with Excel, work with the different options that are in there. I'm not going to go through the step-by-step process of this for both Excel or for both Mac and Word, or Mac and PC computers, and Chromebooks and so on, because everything is formatted just a little bit differently, that it's not going to be that useful. But let's talk about very quickly how to create a bar chart from our data. So I'm going to stop the share here, swap over to our Excel file again. And I'm going to delete our pie chart just by highlighting it with that light blue edge bar. And now let's create some bar graphs. And I'm going to create, I think, vertical bar graphs here. So we'll come up here. Let me talk you through. We've highlighted the whole section, all the data that we want to highlight, which is in this case, well, let's not do the control, let's do the experimental. So we'll highlight just the data labels, which are the average, maximum, and minimum, as well as pretest, post-test, and difference. And then the mean values and max and min. Now, we come up to the chart feature again, and it says insert chart at the top. And if we come to column, we can I'll do the simplest one, the top left one, the clustered column. or maybe we want to change it to a 3D clustered column, or maybe we want to change it to clustered cones because we're a little weird that way, or what have you, right? So there's lots of different options. Maybe we want to do a stacked graph to show the relative sizes all together of the maximum values, average values, and minimum values. That doesn't seem as informative to me, so I'm just going to do the clustered one, just a straightforward one. And so we can see that in blue is the pre-test values, sort of a reddish color is the post-test values, and then the difference is in green. And you can see that the post-test values for the average were higher than the pre-test values, that among the maximum, they were all the same. They all went to 5. The minimum, the smallest one, was in the difference. Not surprising, because minimum for pre and post test could only be 0. Right? But nonetheless, this is a real quick and easy way. And if you want to do different things with different formats and layouts and styles for this too, you can come up to those same 2 bars at the top, the chart quick layouts, and put in the title, maybe label the charts, the bars inside. Maybe you want to do it the default way, maybe something new with showing the actual data points. It's very useful very frequently. Maybe we want to have the chart below of the actual data included with the bar graph itself. Lots of different things. And again, same up top here, we can change the colors. And if you click the down arrows, there are more options than you will ever need available here. So maybe we'll click that one, right? Choose a background color. And all of these can be edited by clicking into the cell and then usually right-clicking on the different fields to change the chart title, double-click, highlight, new title is what we'll call that one. And all of these are ways that you can edit the format for your bar chart. Okay, so lots of neat things. Excel is really a useful program for doing all of this kind of work. And then you can also, of course, modify things. If you want to try this out yourself, feel free. Take your data set, create a bar chart, whether it's a horizontal one or a vertical one, choose it and go with it. There are lots of fun things to play around with here. Okay, if you'd like to do it now, you can freeze the screen and then continue on when we're ready to go. All right, so a thing to think about when we're presenting charts, if we're, in this case, showing 2 sets of vertical bar charts, and they're for 2 different questions. On the one side, it says Grizzly Ridge, the name of the habitat or the exhibit at this zoo, enhanced my appreciation for Ohio wildlife. And then again, it shows the number of samples. And then there was a separate question that said, Grizzly Ridge inspired me to spend more time outdoors. Same sample size. And then we have the percentage of responses that answered either strongly disagree, disagree, neutral, I'm not sure, agree, or strongly agree. For each of these, what we would like to make certain that you do and emphasize that when you're showing your descriptive data, if you're trying to make a point of comparing these 2 pieces of data, these 2 items, make certain that the scale on the y-axis here is the same. Because if you have this being maxed out at 60% at the largest value on the screen, and this one maxed out at 100%, it'll make these agree and strongly disagree, which are much more common than inspiring me to spend more time outdoors. It'll shrink them down. So if you're trying to compare 2 different items that are very similarly worded, have a similar kind of setup, like here with these 2 Likert scales, make certain that you have the scales consistent from one chart to the next chart. Okay? Just a couple quick best practices on summarizing your descriptive data visually. Okay, so try that out. Try taking 2 items that are worded very similarly in your survey. If you have data already that you've already started creating, fantastic. If not, just make something up and try coming up with 2 charts and experiment with changing the axes, because you can do this very easily by right-clicking on the axis and change what is the maximum and minimum. And let me show you how to do that very quickly on our Excel file. So we've done that here. We have our vertical axis. We can, oops, not do that. Control Z is my favorite key. So if we want to change the format, the axis, we can click on that and then have the scale where it says here the maximum is 6, and you can see that's the highest one. Maybe we want to change this maximum to be 10 across 2 different questions that we're asking. So this is on a scale of 1 to 10 in this hypothetical example. No one here gave us a value greater than 5, but we want to be able to compare it to another chart where there are answers greater than 10. So in that case, you would change the maximum just by clicking into the cell on the scale, as you can see on the screen, and changing it to the number value that you want to be the highest or the lowest or whatever it is that you would like. You can also change the major units, which is where there are horizontal lines as dividers and so on. Okay, so I'm gonna cancel that. That's a useful tool. And we'll come back to our PowerPoint. Make certain that the scales are consistent. And same thing, as we had said earlier, make certain that you're including the sample sizes on them. Okay, so we've been talking about descriptive statistics. Now, let's talk about inferential statistics. Because for many people, this is statistics. If you're talking about doing a statistical analysis, you're doing inferential statistics very frequently. If you're going to do a chi-square test, if you're going to do an ANOVA, if you're trying to determine whether one group is different from another group, or whether 4 groups are different from each other, and if so, how. Okay, so in this case, if we want to know whether these 2 groups on the left are different from each other, we have to evaluate our null hypothesis. And hopefully you'll remember that we had a discussion, this is from session 2, about hypotheses. And of course, the null hypothesis is the hypothesis of no change, the H sub 0. Right? Which is that there's no relationship between, or no relationship between this action and this consequence, or there's no consequence of doing this intervention with the restaurants, going in and talking to those different restaurant owners to try to get them to close their dumpster lids, had no effect. That is the null hypothesis. If we saw no difference between the pre-test and the post-test mean values in the experimental group, that supported the null hypothesis. In our case, the post-test was much higher than the pre-test for the experimental groups, significantly higher. And so as a consequence, we would reject the null hypothesis. And we did. Then we also have, of course, our alternative hypotheses that we talked about, that we summarize with H subscript A, H subscript B. And they can be phrased however you want, that increasing values of the independent variable will increase the dependent variable, or same thing, but the opposite, increasing values of the independent variable will decrease the dependent variable, right? So the only difference in those 2 statements is whether the increasing levels will increase the dependent variable or decrease the dependent variable. So we're bringing up hypotheses again, and especially the null hypothesis, because you'll probably remember that it was what we said was the most important hypothesis. And the reason it is the most important hypothesis is that we have to reject the null hypothesis. Okay, so we'll come back to that in just a moment. But when we were talking about hypotheses, we also talked about outcomes. And we talked about what would it look like if the null hypothesis was supported for our question, meaning there is no impact, there is no difference. Right? And in contrast, what would happen if there was a difference? If it led to a preferred hypothesis, what would the data look like? If it looked if it was going to fit an alternative hypothesis that is not our preferred hypothesis, what would the data look like? Okay, and what the data looks like are the descriptive data and the graphs and charts and pie charts and things. So when we're doing statistics, we have to evaluate the null hypothesis, the H sub 0. And in this case, in these three kind of fun little infographics that I found online as an example, that the infographic or the null hypothesis for the first infographic is that cats show no preference for food based on the shape of the food, whether it's fish shaped or triangle shaped. Or plant growth is not affected by light color, whether it's yellow or red. Or the age has no effect on musical ability, young versus older. Okay, so that's the null hypothesis. And what we are trying to figure out is if we can reject the null hypothesis. Because if we can reject the null hypothesis, then we move on to our next hypotheses, our alternative hypotheses. So how do we test whether we can reject the null? Well, we do an inferential statistics test analysis. We run a chi-squared, we run an ANOVA, we run a t-test, we run something. And then we figure out whether the level of significance, which is usually summarized as a p-value, for the probability is what p stands for, the probability value that the results that we got would happen in less than 0.5% of the, or 0.5 of the time. So 5% of the time or less, we would find a significant difference by chance, meaning 95% of the time or greater, we are going to expect a difference, a real difference there that is based on fact. And it is because of the thing that we're trying to test, right? So if we can reject the null hypothesis, we can then move on to our alternative hypotheses, including our preferred hypothesis. All right, so inferential statistics allow us to determine whether we can reject the null hypothesis and then move on to whether the data support the alternative preferred or the other alternative hypothesis. Make sense? And that's why the null hypothesis is the most important hypothesis that we have. Okay, so let's continue on here. If we were to report this data and be in a science magazine article like this, and we're reading through the results of our study and we're finding statistically significant data and summary information that tells us whether things are significantly different from each other, as you can see just above my fingers here. You see where that p-value is. It says p is less than 0.001, two-way ANOVA with post hoc multi-comparisons test. That's the name of the test. And the p less than 0.001 is the p-value. Okay, so if the p-value is less than 0.05, and 0.001 is certainly less than 0.005, then we would reject the null hypothesis. So similarly, here's one on the screen, and that's a brand new science that just came out today that I just got in my mailbox. So here's a quote that I found online, that there is no difference in male and female proportions between the control and treatment group. And it tells you what the test is, chi-squared test, the degrees of freedom, the number of groups that you're comparing, basically, which is the number of groups minus 1, and that's your degree of freedom, so 1. So they had two groups that they were comparing, male versus female. Those are the two groups. They tell you the chi-squared value, in this case 3.636, you take that value and look it up in a table. Now you don't have to, because R will tell you that value as well as the p-value, right? So in this case, if we look at just the p-value here, 0.057, can we reject the null hypothesis? The answer is no, because 0.057 is greater than 0.05, right? Just barely, just 7/1000ths of a probability, meaning 0.7% of the time, more than the 5% of the time. So as a consequence, we would not reject the null hypothesis here, even as close as that p-value is, because we have to be hard and fast. This is what the statisticians have decided is the most useful number based on probabilities, 0.05. So that's what we use, usually. There are exceptions to that, but they are not common. So if you're analyzing your data correctly, p less than 0.05 means that you reject the null hypothesis. So if instead of 0.057 here on this screen, our result had a p-value of 0.0057, would we then reject the null hypothesis? The answer is, of course, yes, because 0.0057 is less than 0.057. Instead of 5% of the time, it's 0.57% of the time, which is a lot less than 5% of the time. Make sense? So that's a way to be able to tell from your brand new science magazines or your research articles that you're reading whether someone found a significant difference between two groups, right? So if we go back up here and their original results had a p-value of 0.057, we conclude, as they did, that there is no difference in male and female proportions between the control and treated group in their perception of something. Make sense? Okay, so we're getting into it here. We're into the descriptive statistics. So what we do here when we're doing statistics is once we find the test and we run the statistical test and we analyze our data in ways that we'll get to in a moment, we go into hypothesis testing. If the p is less than 0.05, we say that it is significant, and we say that the null hypothesis is rejected. And if we did a treatment, we say that there is an effect of the treatment detected, as it says on this part here, or that there is a difference between two groups, male versus female, rural people versus urban people, people with permanent crawls, people without permanent crawls. Okay? Then, how do we know what to conclude? Because our p-value tells us that there's a significant difference, so we reject the null hypothesis. How do we know whether we can accept our preferred hypothesis or our alternative hypothesis? If we just keep it simple, having only two, preferred and alternative. Well, we have to look at the data. And what we can do is look and see whether the control group mean value or average value is greater than the average value, that's how you summarize mean or averages, than the experimental group. If that's the case, then the treatment decreased the dependent variable, right? If the control group was greater than the experimental group, it decreased the dependent variable, whatever that was. If in contrast, like in our experiment with the ravens in restaurants and closing their trash bin lids, that the mean value of the experimental, which we calculated, is greater than the mean value of the control group, which was the case, then the treatment increased the dependent variable. Right? And so if we go back to our data here for our ravens and restaurants, we see here in the experimental group that the post-test difference was 3.8, whereas in the control group, it was 2.8. An entire event greater. One more time closed, almost 4 out of 5 times in the experimental post-test restaurants, the lids were closed. Whereas in the control group, almost a little less than 3 times out of 5 were they closed in the post-test time, right? So we would conclude from this in a similar way to what we just did with that last hypothetical example, that our treatment increased the dependent variable. Make sense? And that, my friends, is how you use statistics to determine whether groups are different from each other, whether our treatment had an effect, what have you. Fascinating, right? And that's how you use statistics. And that's why statistics are some of the coolest things on the planet, because it allows us to say that we had an impact and it was significantly different in our experimental group versus our control group. That's exciting for me, at least, hopefully for you too. In contrast, if our p-value was greater than 0.05, which we saw in that previous screen, which I cannot seem to get back to here, then we say that there is no difference between male and female proportions between the control and treatment groups, right? That's what happens when p is greater than or equal to, note the horizontal line underneath the greater than sign. If p is greater than or equal to 0.05, we say that the null hypothesis is supported. And that's where we stop. We don't look at our alternative. We don't look at our preferred hypothesis. Because if the null hypothesis is not rejected, if it is in fact supported, then there is no effect of our treatment or no difference between the groups. This is a great illustration. I found this at this website here, thebiologyprimer.com, Introduction to the Scientific Method. They have several really nice visual summaries of how to do science, how to do statistics. So I'd encourage you to check that out. Okay, so continuing on then. We're in inferential statistics. We're trying to determine whether there are differences between groups, whether this fourth group here on the screen is by percentage greater than groups 3, 2, or 1, right? And to do that, we use statistical tests, those same ones we've talked about several times, ANOVA, t-test, regressions, chi-squares, a whole bunch of non-parametric statistical tests, which I'll talk about what those are in just a minute, including the Friedman's test. But we use these to tell whether we can reject our null hypothesis. And that's why the null hypothesis, yet again, is the most important hypothesis, because it is the one that we have to first address and that we are testing with our statistics, with our inferential statistics. Yeah? Okay. So we're going to get to how do you choose your statistical test in just a minute. And I'm going to share with you a bunch of different tools to go about making those decisions. But before we can do that, we have to know what kind of data we're trying to summarize, or what kind of data that we have, right? And this is thinking about both our independent variable and our dependent variable. So if usually we are analyzing our dependent variable, of course, because our dependent variable is responding to the independent variable, our dependent variable is dependent upon the independent variable for its outcome, right? Or if you remember, another form that we use often in social sciences are that the response variable responds to the predictor variable. The predictor variable predicts how the response variable is going to manifest. So you'll have different kinds of data in your data set, in your Excel file. Some will be nominal, or what we call categorical as well. And this are data that have categories. There's not a logical way to rank them or hierarchicalize them. So examples of this could be gender of your participants, could be ethnicity of your participants, where they live, their residency. Often categorical questions, yes, no questions. Are you a farmer? Are you not a farmer? These are categorical data. So if you have categorical data that you're trying to analyze, you use the right test. And our ways that we're going to choose between our different statistical tests will depend on whether it is nominal, categorical, or these other two as well. Okay, so that's categorical or nominal data. Use those as kind of interchangeable with each other. Second kind of data that we can have is what's called ordinal or interval data. And these data are not necessarily not predictable in the intervals between the data. There may be a logical way to rank them. Like, for example, Likert data, strongly disagree, disagree, neutral, agree, and strongly agree, right? So there's a logical ordering to it. But what I mean when I say strongly agree might be very different than what you mean by strongly agree, right? So similarly, my agree may have a greater difference between agree and strongly agree than my agree does with its neutral. So the intervals between each of those 5, which we as experimenters assume are the same, may not be the same, right? You could think of a situation where we have 5 categories, and instead of being equally dispersed in the mind of the participant, one of them is so much different than the others that it is off on its own. And these 4 are very clumped together. Make sense? So this could be strongly disagree, strongly disagree, disagree, neutral, agree, and strongly agree. So they might think the difference between strongly agree and agree is so much greater than the difference between any of the other groups. So Likert values, Likert items, rankings. I might really like one, but not really care too much about the others. So it might be hugely different between my top choice and my lowest choice, or even my 4th choice if there's 5 options. Schooling is another example of an ordinal or interval data that is not necessarily the same thing. The amount of information that we learn in between 4 grades in primary school could be a lot more or could be a lot less than the content that we learn in 4 grades in secondary school, than what we learn in 4 grades in postgraduate college. So schooling, ranking, Likert, sometimes ratings, and we're going to get to that in a minute. They may be able to be put in a sequence, in a logical order, but it doesn't necessarily mean that the intervals between each of those options are the same, and that they are thought to be the same by every participant that you have, right? So we analyze these in a different way, these ordinal or interval ones. Usually these are done with non-parametric statistics, to be honest, if you know what those are. If not, we'll get to those in a second. Our third kind of data is what we call numerical or continuous data. These are data that have very clear, regular intervals between data. 1 is 1 more than 2. 2 is 1, I'm sorry, 2 is 1 more than 1. 3 is 1 more than 2. 4 is 1 more than 3. 5 is 1 more than 4, and so on, right? So the interval is the same between each of the 5 points. Same thing with heights, right? 200 centimeters is 10 centimeters less than 210, which is 10 centimeters less than 220, and so on, right? Their continuous or numerical data are a predictable intervals between each of the data points. 1 year, 2 year, 3 years, 4 years, 5 years, so on. So when the intervals are consistent between each of the possible data points, we're talking about numerical or continuous data, okay? And only with continuous data or numerical data, again, same thing, are averages a real thing. So we could put up an average of the Likert scale, average Likert scale being 3.4 out of 5. But we have to acknowledge that the difference between that interval data or that ordinal data, same thing, you'll see these 2 terms being used interchangeably or differently on different screens, different websites that we'll get to in a second. So we can come up with averages, means, for Likerts, rankings, schooling, and so on, but it's not necessarily accurate to call them means or averages. It's better to say the most common version, right? Most common Likert value received was 4, that kind of thing. You can use means, but often it's suspect. People use it all the time, but if you do statistics on it, you have to make certain that you use the right inferential statistic test. Okay, so think about these three kinds of data: nominal, ordinal, numerical, or categorical, interval, continuous, because we're going to come to those in just a moment. There's a great website that's available at the bottom of the screen here from the University of California, Los Angeles, which is what UCLA stands for, that has a really nice description between categorical, ordinal, and numerical variables. So check out that website. It'll go through in great detail talking about all of this. And I'm going to switch over to that screen right now because it's a very useful program, a very useful summary. And that is here. So that's what this is. What's the difference between categorical, ordinal, and numerical variables? All right. Read through this. It's a nice description, very brief, about what each of these mean and what are the consequences of whether a variable is categorical, ordinal, or numerical. All right, or whether they're intervals or continuous data. So we'll come back to our PowerPoint now. Thanks for your patience as I swap between all these different slides, different modes today, but it's very important. So now that we know what kind of data we're trying to analyze, now we can go to some resources and figure out which statistical test, which inferential statistics test we can use. Okay, there's 4 different choices on the screen here. I'm going to talk through 2 of them, and I'm going to walk us through 2 of them, the second and third ones. So layered statistics, it's a great program. If you have the resources to be able to buy a premium membership to Layered, L-A-E-R-D, that's how you pronounce that word, that name, Layered. It's a great resource. It's the best resource for statistics that I can find online. And most of my friends agree that's what most of us use. So check out Layered Statistics. I think it's around $70 or $80 US for a year's membership, and I use it all the time. So it's well worth the money for me. The next 2, UCLA Stats. This is another website from that same program at UCLA. And we'll come back to that one because it has a nice choice test box, a grid of options that we can use. And this website that I had never heard before called Simply Educate Me has this really nice, probably my favorite of the 2, of UCLA and the Simply Educate Me, about statistical analysis, how to choose the appropriate inferential statistic test. And then last is a dichotomous statistical choice key that I've created in 2002, I think it was, for a class that I taught to undergraduates. Worked really well for them. They were able to go through and figure out what they want. And I'm going to have this available in our group, in our class group online under the resources topic. So please feel free to walk through, check out all of these, especially the last 3, which are all free. The Layered Statistics, if you can subscribe to that, that's the best. That's the premium. If you don't have the funds for that, then let's do one of these other 3. Okay, so I'm going to go to the UCLA Statistics site first, and then I'll go to the Simply Educate Me thereafter. Once I can find the thing I'm trying to find. There it is. All right, so this is what Layered Statistics looks like online. It's a really nice site, has lots of options. It will walk you through, especially if you use SPSS, how to do every single statistical test you can think of. You can see all of them listed out on the board here. Most of what we've been talking about are that column all the way to the right side, differences between groups, and that's what we're going to stay focused on for most of the rest of today. But also, of course, predictions and associations between 2 variables that we don't necessarily have a clear reason for why one would have influenced the other, but there seems to be an association. And then a bunch of other possible options. Layered is a great program, a little pricey, but worth the investment if you can afford it. Now, this next one, the UCLA Statistical Consulting page has this series of options. Now, we're talking about the number of dependent variables, number of independent variables, and then the nature of those dependent variables, whether they're interval or normal, or what we are calling continuous or numerical, whether they're ordinal or interval, and categorical. So we have different things, and if we know how many dependent variables we're trying to analyze, usually we're just looking at one, right? And the number of independent variables that we're trying to use to predict what that dependent variable is, we can go through and, based on the nature of the dependent variables, whether it's an interval, an ordinal, or a continuous variable, we can then choose the proper statistical test. And one sample t-test, let's see if we go from one variable to one independent variables with two independent variable levels. So male versus female, for example, and we're trying to determine their preference for wooden cook stoves or sustainable cook stoves. We would use a two independent sample t-test, because that would be an interval basis for the dependent variable, which is a preference. Okay, so this is a great site. Spend some time with it. You can play around with it. We're going to come back to the most common statistics, and I'm going to walk us through doing some of those statistics on R in just a moment. But choosing the proper statistical test is really important. Similarly, this is the Simply Educate Me website that has a nice way to choose a statistical test using the same thing. What kind of data is it? Is it an interval? Is it an ordinal value? Or is it a categorical, a frequency, or what we call nominal, right? Male versus female, for example. Ratio or interval would be more of the Likert scales, and ordinal would be more like a continual data. So we have these different options. If we have the type of data, the number of groups we're trying to compare, male versus female, ranchers or farmers versus not farmers, and whether we're trying to test for a correlation between two variables where we don't know that one has definitely led to the other, but they seem to change in the same way. Whether we're looking at variances, differences between different groups in that way, looking at just comparing means, whether it's an association or a correlation, we can use these specific tests, right? It's a pretty useful tool, this Simply Educate Me one, but you'll notice that the number of types of statistical tests and the number of them is quite limited, particularly relative to the UCLA test. The UCLA one is much more robust. The Simply Educate Me one is a little smaller, but it works for the data that it summarizes. Okay. Last one that I want to share with you is the one that I have created, which is shoot, do I not have that open? I think I might have closed it. No, here it is. Is a program, a dichotomous key that will allow us to choose the proper statistical choice test, and I seem to have not opened it. So I'm going to have that available on the website, and we'll come back to that screen in just a moment. But it's a useful dichotomous key. I like it a lot. It's the one that I use most commonly, really. Biased because I made it, but also because I think it's the most useful way to come to statistics and the most clear way to choose your inferential statistics test. So please check out the dichotomous key that I have that is under our resources tab topic, rather, on our Facebook page. Okay, so the most common types of inferential statistics that we use are really these 5 for most of us. And for many of us, we just use the top 3, the chi-squared test, t-test, and ANOVA, right? A chi-squared test is a categorical dependent variable, right? Male versus female, farmer versus not farmer, people who participated in the group versus people who did not. So for those tests that I was showing you about our Ravens and restaurant tests with closing the rubbish bins, this is what we used, was chi-squared test, because it was a categorical dependent variable, which is the number of times that the restaurant closed their lid, 1 through 5. And the one independent variable with 2 levels, which is control group versus test group versus experimental group. So we use chi-squared test for that. Similarly, you can use t-tests. If we had looked at the number of, the weight of garbage that were in each of the bins for whatever reason, we were interested in that. That's a value that would change from no trash whatsoever to thousands of kilograms inside a single dumpster, because they're very large. For that, if we had the one independent variable, which was whether it was a treatment group or not, and then we had a continuous or numerical dependent variable, which would be the weight of the trash. In that case, if we did the intervention and we weighed the amount of trash that were in the bins, because our intervention was trying to get them to reduce the amount of trash that they produced, just making things up here, we would then use a t-test. Okay? And if in our case, where we measured their preference or the amount of kilograms of trash that were created before our intervention and afterwards, and we kept track of the same restaurants, so we know what their weight was before and their weight was afterwards, we would use what's called a paired t-test. Going back to what we started our discussion today with was anonymity versus confidentiality. If we went the confidentiality route, where we wanted to talk to people and do an intervention with them before and assess their perspectives on things before versus afterwards, we could do the same thing here, because we have assured confidentiality. We've told participants, that we will not tell others how they responded, but we will know them so that we can go back to them afterwards, after we do the intervention. So we can compare their pre-test data to their post-test data. That's a t-test. That's a paired t-test, even more to the point. Similarly, if we have ANOVA and we have a lot of groups, 2+ levels, so similar to t-test, we can do continuous numerical dependent variables, or also ordinal or interval variables, like our Likert tests, our Likert items. If we have multiple groups within our ANOVAs, say we have people from 3 different communities and we're trying to compare them, those are our independent variables, 3 groups. And then we have the dependent variables, their perception and support of some conservation project. So we could measure those measures of support before and after an intervention, or maybe we're not even doing an intervention, we're just comparing the 3 communities one to the next. We would do an ANOVA. ANOVAs are very commonly used. It's a great way with both ordinal or interval data as well as continuous or numerical data to be able to assess your data points for significant differences. Okay. Friedman is a kind of non-parametric test that is really, really useful, particularly for data that are not necessarily distributed normally. They don't have that bell-shaped curve on them. And usually we have to test for that. And if that's not the case, then we use a Friedman test instead of an ANOVA. Okay? And then multiple regression are when we have continuous data in both the independent variable and the dependent variable. So say we are looking at health of people and the amount of time that they spend exercising, measured in minutes for the amount of time that they exercise and the number of cigarettes that they smoke in a week, say. Those are both continuous values. So we can measure the number of minutes because 1 minute is 1 minute less than 2 minutes, which is 1 minute less than 3 minutes, and so on. And same thing, a cigarette is 1 cigarette, 2 cigarettes are 1 cigarette doubled, 3 cigarettes are another cigarette added in, and so on. So it's a continuous value in terms of the number of cigarettes that people smoke. So if we are looking at the difference between, or the impact of exercise and the number of cigarettes that people smoke, just making things up, we could regress those two numbers, one on the next, create a scatter plot and fix a line to it and determine whether it's significant. And we can also, with regressions, look at multiple independent variables. So it's not just the amount of exercise that people do, but it could be how healthy they are or what have you. It could be a bunch of different variables. So these are 5 common tests that a lot of people do quite a bit. And on the top right of the screen, this is an output from a statistical package. This looks like an SPSS program, but the outputs be very similar to R, as we'll get to in a minute. And this is how you report it. So you can see how they've talked about it on top, that differences in scores for caffeine and no caffeine were significantly different between these conditions. And it was a t-test that we see here. And we can see that it was a paired samples test because that's how it was. It tells us that. And this is the value for that t-value of 5.66. So they rounded up from 5.657 with the degrees of freedom of 4, meaning that there were probably 5 different groups that they were comparing one to the next. And then the significance of this was p equal to 0.005. So can they reject the null hypothesis here? The answer is yes. Right? So this is how outputs would often look, and this is how they're reported in the literature. So now, what do we do with this? We have all these, we've gone through this stuff. You've taken some time, you've studied those statistics choice tools that we talked about, that we addressed and discussed through a little bit on the websites, the dichotomous t, maybe you have a subscription to layered statistics or not. But what do you do with them? So now you want to do the statistical test. What I would recommend to you, if you do not already have a subscription to kind of expensive statistical packages like SPSS or SAS or Minitab, or there's a bunch of others, is to look at R. R is a free computer program that does pretty much all the statistical analyses that you would need to be able to do, and it is freely available. In addition, how R is set up, you have to type in all the commands. So you need to know quite a lot about programming. However, there's another program that you can run at the same time as R called R Commander that becomes what you're actually interfacing with, what you're clicking on and what you're typing numbers in and doing analyses with. R Commander is a point and click kind of setup. So it's very similar to how SPSS or SAS or Minitab or all those other statistical programs work, but it is also free. So R Commander is what you interface. It's the graphical user interface, if you're familiar with that GUI or GUI term, that you would use to run R. It's great, it's free. And R, fortunately, I've created a document, another document that I would like to share with you for free, of course, is that I've created this thing in 2016, about 4 years ago, of using R for statistical analyses. And in this file, which is going to be in our class site, it shows you how to download and install R. It tells you where the website is to go to, how to install it, how to run it, how to import data from an Excel file, which we'll do together here in just a moment, how to choose different tests, and also some additional suggestions on how to do inferential statistics. So it's a good tool. It's really a useful thing for sort of further on, more in-depth study about how to get set up to be able to do the inferential statistical packages and inferential statistical tests. Okay, so we're equipping you with the tools here. We're not necessarily spending a lot of time going through and teaching you how to run the tools. But the resources that I'm providing to you will have that information in them. In this case, this Facebook group post does this. It's a PDF file. So let's go to that R. Let's go to R. If I go directly to R, this is what it looks like. You have to type in all these codes, and the output is very clumsy and hard to read. Here you go down here. But instead, what we could do is I'm going to stop this share, and I'm going to switch over now to R Commander. And R Commander is here. It's run on the Mac on a program called X-Quartz. And it is all done by pointing and clicking. So you come up to the menu and you choose your statistics. Say I'm going to do a means test, a t-test, a paired t-test, a one-way ANOVA, as we've talked about in here. There's also our non-parametrics test, like our Friedman's rank sum test, our dimensional analyses like factor analysis or principal components analyses. These are higher level multivariate kinds of studies. Cluster analyses, which are a fun kind of statistics as well. Our fit models, under which is where we put linear regressions because we're trying to determine whether our data fit a model. And that model is determined numerically using slopes of lines, whether it's a flat line, whether it maybe is a parabolic line or a reverse parabola. And you can choose the statistical program based on that. It's very simple, and it's all done using the point and click. It does have some graphing features in it in R Commander, but they're pretty limited. I wouldn't recommend that you use these options very much in here. I would make my scatter plots, bar charts, pie charts, and whatnot in Excel because the colors are more easily modified and the graphs tend to look a lot better that way. All right. So if we wanted to, how do we bring in data into this? Well, we come up here to the menu bar and we click on the button Data. And we say import data because we're importing it from an Excel file. You can also hand type it into R yourself, although I really recommend that you put your data into Excel. It's so much easier to use. So if we want to import data, we come in here, we go down to From Excel File, and I'm going to choose that same program that we were just talking about and just using, the Ravens and Restaurants data. So the first row in each data is going to be the name of the variable, as we have on here. And this data set is just going to be called data set. And it will be available for the R to be able to analyze. So I'm going to go to say OK. You can also rename this as whatever you want. Raven and Restaurant. Computer is moving very slowly at this moment. And whatever you put in there, you would then hit OK. And it will allow you to import data. It takes you to your program, and you navigate to wherever you are on your computer appropriately. You can see it's sort of an old DOS setup, if you know what DOS is. The data that we were using was from this file right here, so I'm going to click Open. And if it has lots of different sheets in it, you can choose which sheet you want. So we're going to use the second sheet that we created for R, the statistical programs, and I'm going to say OK. And then it's brought it in. And so these are the data that we have. You can see location, experiment or control is the header for the second variable, pre-test values for the restaurants, post-test values for the restaurants, and then difference of pre-test subtracted from post-test. If it's a 0, they stay the same. If it's a positive value in difference, they've more frequently closed their dumpster lids. Now, so we have our data in. That's how you import data into Excel or into R using R Commander. Now, if you wanted to do statistics on this, let's do a chi-squared test. And in here in R, a chi-squared test is called a contingency table. So that's how it is. We're going to do a two-way table, and we're going to put in the two variables that we have that we are interested in. Which should be pre-test and post-test, and they're of course not showing up when I need them to because that's how things work. So we're going to do instead, let me change this. We're going to assume that these values are not in fact categorical values or ordinal data. We're going to call them continuous data because we've been analyzing these numbers of restaurants or these number of times that their lids have closed for a very long time. So we're going to do a t-test, and not just any t-test, but because we know the names of the restaurants, we're going to line up the pre-tests with the post-tests. So we're going to do a t-test here, paired t-test, and we're going to have the pre-test be one variable and the post-test data, pre-test and post-test data, be the second variable that we're going to compare one to the next. And it's going to compare between each individual, before and after, and it's going to line each of those up. So it won't just take an overall mean value and compare it to the overall mean value of the post. It's going to compare restaurant by restaurant to each other and then summarize all those up. So I'm going to say, okay. And this is the result that comes out here. Right? You can see that the label is paired t-test, so you know that's what you've done. The t-value of minus 2.9974, our degrees of freedom are 58, meaning that we had 60 restaurants, take away 1, and 2 groups, control and or pre-test and post-test. So take away 1 again. That's just how you calculate degrees of freedom. And our p-value, as we can see here, is 0.004, 0.004 repeated. So can we reject our null hypothesis that the post-test was different than the pre-test? The answer is yes, right? And what we find is that because we've looked at the averages, we had them on our Excel file, which I'm going to switch over to now to compare the pre-test and the post-test. So our overall, this is the data that we were just looking at for doing the statistics. Our overall average pre-test was 2.69 times when we visited out of 5 the restaurants closed their dumpster lids. Post-test, 3.32. So there was an increase. So we go back to our statistics, back to R, and we had seen that the p-value was significant. So we can reject the null hypothesis, and then based on what we found on our Excel file, that the post-test value was higher than the pre-test value, we can go with our preferred hypothesis, which is that all of our efforts of going to all the restaurants and asking them to cover their trash were a success. So we can do this, of course, with sub-selecting from within it, from just looking at the experimental restaurants, one to the next, and we can screen out the data that doesn't fit that condition. And that's all on Edit Data Set, which you can do and learn how to do on your own. But just on a quick overall thing, looking at pre-test to post-test, we got a significant difference. Hooray! So this is a quick way to learn how to use R. And I'm going to switch back to our PowerPoint now. And that sheet there, that PDF, talks through all the steps that we just did very quickly. How to install the program, which we didn't talk about. How to install R Commander, which we didn't talk about. How to make R Commander start R. And how to do statistics. So it shows you all of what we just did and in much greater detail. So please make certain to take a look at that file that I've created and made available for you. So if we want to be able to answer our questions, can we reject our null hypothesis or not, we have to use inferential statistics. Descriptive statistics, as we show on the screen here, are not enough to be able to say that our rearing option number 5 was significantly greater than rearing options 4, 3, 2, or 1, right? We have to do the statistical analysis. We have to determine if we can reject the null hypothesis, and if we can, which alternative hypothesis can we support, right? If rearing option 5 was something very different, maybe it's rooting these coral in the wild, and this is a fire coral being rooted into some other aquaphora, some reef building coral, then we can say, yes, we reject the null hypothesis that these 5 rearing options are the same. We look at our data and we see that rearing option 5 was so much greater than the others. So we can do our post hoc tests after that to determine what is different from which. Or we can just look at the descriptive data, and as a thumbnail, we can conclude option 5 clearly worked well, right? So if we look at this, this is how we would go about rejecting the null hypothesis. We have our ANOVA, our t-value of with these kinds of degrees of freedom, a very large value, and the p-value is less than 0.001. So can we reject the null hypothesis? Yes. And then we figure out from the actual data, as we did with the ravens and restaurants data, which of the hypotheses we can support. That's how you do statistics. That's how you do inferential statistics. Sometimes, of course, when we do our statistics, we do our inferential statistics, we don't find a difference. We cannot reject the null hypothesis, right? This is called a null effect. Null meaning 0, like the null hypothesis, 0. And when we get a null effect, meaning that we have not found a significant difference between 2 groups, it's because we cannot reject the null hypothesis. There are a bunch of reasons for why this can be. Often it's because of our experimental design. Maybe we didn't ask enough people. Maybe we didn't interview enough participants. Maybe there's not enough difference between the 2 groups for the variable that we're analyzing. Or maybe there's too much variance between the 2 groups, and it all just kind of swamps out. We're going to get through each of these differences. Or maybe there is no difference at all, and no matter what we do, no matter how many samples we collect, it's not going to come back as significant, right? So these are the 3 reasons why you may get a null effect. So in this one, this is the first one, not enough between groups differences. So we take the average happiness a day later after people get some cash, or I'm sorry, no cash, some cash, a few bills, or a whole bunch of cash. And we find that 1 day later, the same number of people across all 3 groups report the same level of happiness. So it could be that there's not enough differences between each of those groups. Maybe a lot of cash really isn't all that much more relative to some cash. Maybe we're giving out, for the no cash, we're giving nothing, obviously. Maybe we're only giving 50 cents out for some cash, and we're giving $2 or $1 out for a lot of cash. And maybe in that case, our experiment was not designed particularly well because there was not really enough difference between these 3 groups. The difference being the amount of money that we gave to each of the 3 groups. If we instead gave 0 to the no cash, 5 to the some cash, and $1,000 to a lot of cash to those groups, maybe we would see a big change in happiness a day later. maybe not. Okay. Next up, maybe there's not enough between group differences in the sense of we can't really calculate them correctly, right? The anxiety level wasn't really assessed correctly. It wasn't the manipulation didn't cause enough anxiety. High anxiety is not really much of a difference from the medium to the low, like we were talking about with dollars. Another important reason why we may not find significant difference, may get a null effect, is that we have maxed out what's going on, right? So the problems are too easy, for example, so we're getting a ceiling effect. Everyone answers them correctly. Or what you're asking is so objectionable that no one is going to say that's a good thing. So everyone says, no, that's the wrong thing to do, because you're asking them to do something morally offensive. So no one will ever want to do that, right? Maybe in the same way, the problems are too hard so that everyone gets them wrong, or the question is too challenging so everyone gets it wrong. So you're asking them to remember the weather and how their livestock did 10 years ago. People are not going to remember that very well. So the problem responding to the problem accurately is going to be too hard because it's too far away. I don't remember. And so the responses give the wrong answers, and we get what we call a floor effect. Okay. So there could be not enough between group differences. We may not have correctly manipulated the independent variable. Anxiety wasn't high enough. Maybe the dependent variable was not sufficiently sensitive. Maybe what we call happiness, or how we measured happiness, didn't work very well at measuring true happiness. Maybe there's the ceiling or floor effect on our dependent variable, or maybe there's a ceiling or floor effect on our independent variable. Okay, so how we differ things may be too hard or too easy, or maybe how we measure that that test. This GRE is a graduate record exam. It's an exam that we do for allowing people into graduate school in the United States, or at least have in the past. That maybe everyone gets the same amount right, no matter if they've done a very extensive prep course, or they just get a pep talk where they're made to feel better and more confident, right? So alternatively, there may not be much within group difference. So we're not finding much of a difference within each group. In the groups on the left, we find no difference because these bars are a summary of standard error in our data, the amount of variance that we have in our data. Generally, if these whisker bars overlap between two groups, they are not going to be significantly different, especially if they overlap a lot. But if they overlap a very small amount, as in the case on the right in column B, maybe they will be significantly different from each other, the GRE course versus the pep talk. So in the case on the left, on A, there is a lot of within group variance. And it's so variable that we can't really detect a difference. And that's why the error bars are so much greater in A versus when we look at the raw data for B, they are very clumped together, much closer. And so we get a smaller error bars, same averages, right? Same mean values across the two conditions, the two hypothetical conditions. But in the latter case, there is much greater there's not as much within group difference. And so you're going to be more likely to see a significant difference statistically. Whereas here, where there's a huge within group difference, it's going to increase the error bars so much that you're not going to find a statistically significant difference when you do your inferential statistic. Okay? However, perhaps there is no real difference there. right? The two groups that we think are going to be different, the treatment groups and the control groups, are not really different from each other. Possibly because we did a bad job of designing the experiment, possibly because the intervention did not actually make the difference that we were hoping to, possibly because there was no difference between men and women in your groups or your people's support for the stoves, the sustainable stoves, what have you. Sometimes there is no difference, and it's not an experimental flaw. There just really is no difference there. And so I found this online. I like this a lot. Roses are red, violets are blue. If you were a null hypothesis, I would fail to reject you. So sometimes there is no difference, and that's what we get. Not always will you get significant differences between your groups. Not always are your interventions going to be successful and produce a significant difference. We have to be ready for that. And that's the way of doing science. So I hope this introduction was useful to you. And I really hope in particular that you're going to go through and use those resources. The understanding what the differences in types of data are, nominal, interval, continuous, knowing how to choose your statistics tests using those different resources that we were talking about, especially that dichotomous choice test key that I created. Hopefully you will use R and R Commander, and that guide to how to install and use R and R Commander are useful for you. Because if you can do all of these things, you're making a real difference, because you are really doing the statistical analyses, the inferential statistical analyses that will allow you to make big conclusions and to demonstrate to your donors that you're making a difference. And that's the value of statistics. So sometimes we can improve a detection of a difference between two groups that maybe wouldn't be there if we include a control group. Maybe with this Raven and Restaurants study, we hadn't included a control group, and we just went in and did the intervention and saw a difference, or didn't see a difference. But if we had also done the control group like we did, you're more likely to be able to detect a difference that is there if you add that control group, which is the group that you're not doing the treatment with or not doing the intervention with. So sometimes it's good to have a control group because it helps to improve the rigor of your science and the strength of your statistical analyses as well. Sometimes if you create your control group poorly, you might have an out of control group. That's not a real thing. I just thought that was a funny graphic by this comic. So control groups are really helpful to have. If you can incorporate them into your treatment, into your interventions, it's a great way to improve the statistical strength of your analyses. All right? And when I talk about statistical strength, I mean, of course, inferential statistical strength. Okay, so that's a whole lot of theory and statistics and inferential and descriptive statistics and hands-on stuff of making charts and graphs and running a couple statistical programs. Download and install that R and R Commander. It'll make your life better. Use Excel to make your graphs. Use R and R Commander to do your inferential statistics, and maybe also to create your basic data summaries, your descriptive statistics too, that you can then use in Excel to create your graphs. I love quantitative data. It's helped to make me who I am. It makes my science better. It makes my conservation better. And of course, by working with our communities, we're making conservation better. So thanks for your time. Thanks for sticking with us. Next week and the week after that, Alex is going to be here to talk about qualitative data, which is awesome. Qualitative items, open-ended questions. I don't know about you, but I'm really looking forward to seeing those. She's an awesome presenter and she's going to have great stuff to say. So thanks again for helping to make certain that communities matter for conservation. We're doing great stuff. It's a pleasure to work with you all. And I am grateful for the opportunity and hope that you have been enjoying this series and continue to participate. because we're doing great stuff together. See you in a few.